So what we're interested in is, can we use a relatively new technology, single cell sequencing, that allows us to look at the gene expression of cells and kind of identify how cells really, how the transcriptomic profiles of cells is working. And this is in big contrast to existing bulk tissue measurements, where we average the expressions of genes across many different cell types. And the goal is to link genotype data with single-cell RNA sequencing to see the genetic regulation of cell type-specific effects. So essentially what we try to do, our exposure is cell type-specific gene expression, and we want to link it with our outcome of interest. This may be human brain disease or behavior. One of the problems that we have is, obviously, we have confounding factors. And you could think here of typical epidemiological confounders, but also of technical artifacts, like, for example, batch effects. And what we are proposing to do is we want to use genetic variants, in particular cis-EQTLs, as instrumental variables in a Mendelian randomization paradigm. And this so-called two-sample summary level MR paradigm allows us to integrate data from our cis-EQTL study with GWAS, or genome-wide association studies, with the outcome traits of interest. So here is a little bit about a small print about MR. So this is basically the assumptions that need to hold in order for MR to provide valid inference. So the most, one of the first assumptions, the only one that's actually testable, is the relevant assumption. This is essentially this arrow in the stack that says that the genetic variant is associated with the exposure of interest. The second assumption is the exchangeability assumption that says that the genetic variant, the instrumental variable that you want to use, is independent of all of the confounders of the exposure outcome association. And the final assumption is the exclusion restriction assumption. This says that the genetic variant is independent of the outcome Y, conditional on the exposure X or the confounder U. So this means essentially that there's no other path going from G to Y than via X. So often, as also Neil has shown, people think of MR as kind of a natural experimentation, where we have a random allocation of alleles at meiosis, where some part of the population has genotype B, which results in normal LDL cholesterol level, while in contrast, the other part of the population is allocated to genotype A, which has lower LDL cholesterol level and might have a lower rate in cardiovascular events. So coming to the methods, our analysis plan has mainly 4 big steps. The first one is the quality control and data preparation. Then we do the EQTL calling. This is followed by a colocalization analysis to select valid instruments. And the final step is the Mendelian randomization analysis. So talking in more detail about the data that we have. So this here shows a t-SNE plot of the 8 main cell types that we're able to call in our data set. In total, our sample size is 128 individuals, where we have around 4,500 cells per subject. And this makes around 600,000 nuclei across sample set. And as said, we have integrated this data into the 8 major cell types in the human brain. So the next step is to use the genotype data with the single cell expression using the matrix EQTL tool to generate single cell EQTL. So you see the results of this analysis for one example, one particular SNP and a gene. So this is SNX31. And here you can now see the association that we have for the G allele GT versus TT, and how the expression at this particular locus changes depending on the alleles you have. And what you can see here, really nicely here, is we have a really strong cell type specificity. So you see that there is an EQTL, so where there's difference in expression only for astrocytes, where for all of the other cell types, we see almost no differences in expression. So if we would have done a bulk measurement here and averaged across, it's unlikely we would have been able to detect this type of effects. So here is an overview of the overall discoveries that we have. So this is here coded by the 8 different cell types, and this is the number of total EQTLs or E-genes, respectively, that we find in our data set. Here are some more examples for cell type specificity. So for example, this gene OGFOD2, you see that we have a very strong effect in excitatory neurons with a very strong p-values, while for all of the other cell types, there's literally very little effect to be detected. Similar for one of the key results that I'm coming to later, p-calm, we have here a very, very strong effect in microglia only, but not in the other cell types that were part of the panel. So I mentioned already the small print of MR, which often is known as the no pleiotropy assumption. So there could be a pleiotropic pathway A that connects G with Y that doesn't go via X. And this is one of the key concerns in MR study. And lots of the methodology work in MR is about making MR more robust to this type of horizontal pleiotropy. The problem that we have when we do drug target or cis-MR, where we only have one single genetic variant, is that we cannot use the robust MR approaches to take care or model the unmeasured pleiotropy. So the problem that we have here is we have in the region of interest one genetic variant G1 that's in LD with another genetic variant that may act via a different pleiotropic pathway. And here the necessary methodological tool to use is co-localization. And co-localization is a Bayesian approach to test if 2 traits share the same genetic architecture. For here is an example where we have 5 SNPs, 1, 2, 3, 4, 5 in trait 1. Here is trait 2. And co-localization is looking at the evidence for 4 distinct hypotheses. The first hypothesis is that there's only an effect in trait 1. The second one is that there's only an effect in trait 2. The third one says that there's an association only with trait 1, but these are different SNPs. Well, the hypothesis H4 that we're actually interested in says it's exactly the same genetic variant that is causative of trait 1 and for trait 2. So here is an example plot. This is taken from the PCSK9 gene region. We see here local Manhattan plots, the first one for LDL cholesterol and the second one for coronary artery disease. What you see very nicely here, we have strong co-localization evidence for LDL and coronary artery disease, where it's exactly the same top hit that's shared between the 2 of them. Well, for example, for Alzheimer's disease, we see a completely different pattern of genetic associations in this gene region, indicating that there's not the same underlying genetic architecture within this particular region of interest. So coming to the results, here is an overview of the co-localization hits that we have for the different type of phenotypes and outcomes. We find the most co-localization hits for schizophrenia, followed by IQ, and we have 33 hits for Alzheimer's disease. So here, zooming in on Alzheimer's, you see all of the 33 different potential co-localization hits. You see here on the y-axis the different genes that we find, and on the x-axis the 8 different cell types. And what you can see already in this picture is that we have very strong cell type specificity, but there are some genes for which we have co-localization evidence in more than one cell type. So here is, for example, a co-localization plot for Alzheimer's disease and PCAL, where we also have pretty strong co-localization evidence, where it's exactly the same lead SNP that's shared between the expression of PCAL in microglia and the GWAS for Alzheimer's disease. Here is an overview of all our significant MR findings. So you see here the number of hits was largest for schizophrenia and IQ, but we also have pretty strong overlap where we see the same gene to cell type combination being likely targets for more than one disease. One example here is for schizophrenia and IQ. You see here in this plot on the x-axis the beta coefficient for schizophrenia, the beta coefficient for IQ. And here you can see the pattern, how the gene expression links between those diseases here. Zooming in a little bit in our key findings, I mentioned already PCALM expression in microglia for Alzheimer's disease. PCALM is known to modulate Alzheimer's disease risk via clearance of alpha, beta, and tau. And it has been shown that a reduction of PCALM expression increases tau decomposition. And it's suggested that PCALM activation may act as a potential therapeutic target strategy for Alzheimer's disease. The next one, EGFR, it has been fine-mapped in the Belengues Chivas on Alzheimer's in 2022, but there was no data on the directionality and what is the kind of cell type that it's associated with. Also, EGFR inhibition reduces alpha-beta tau pathology and reactive astrocytes in several models of AD, and there are new blood-brain barriers penetrant EGFR inhibitors. For Parkinson's, we also have one very interesting example. It's GBNMB. And here the activity results in loss of cellular internalization of fibrillar alpha-synuclein and reduced PT pathogenicity. And levels of GBNP are correlating with PD severity, so it could also serve as a potential biomarker. And it suggests that GBNP inhibition as a potential therapeutic strategy for Parkinson's disease. So here this is, in this plot, you see our 16 Alzheimer's disease hits that we have from MR, where you see here the effect size on the x-axis, either indicating activation or inhibition, so kind of which direction the target would need to act. And here in red is highlighted expression of genes that have potential drugs linked to them, so that could be used to take forward for prioritization as drug targets. So coming to the conclusion of this work, so the principal we present here principal framework for single-cell Mendelian randomization target discovery in human brain phenotypes. One important aspect is that the data generation was on healthy individuals, and the team has made a big effort to look and ensure that the data sets, the samples, are really pathologically healthy individuals, which is quite a big effort given that we talk about brain samples here. This is an important step to rule out or reduce the risk of reverse causation, as we've seen what Neil has done. What Neil showed about the problem that also a disease may cause changes in the phenome. And here we want to ensure this by measuring the expression data on healthy individuals only. In total, we have genetic evidence for 118 genes across 23 human brain diseases and behavioral traits. We also find 21 genes that contribute to more than 2 brain phenotypes. And this approach that we've done also provides us a direction of the relationship. This informs the therapeutic development. And it allows us to define causal mechanisms and also cell types of action, which will help to the development of drug targets. And this can inform how to tailor preclinical experiments. As the next steps, so as I mentioned that we had 21 genes that affect more than one phenotype, we're also interested in looking into multi-response model. We have recently proposed the first MR model for looking at more than one outcome. And here the idea is to model the relationship between more than two outcomes. And also it allows us to define shared and distinct causes of disease. The other methodological development that we want to look at are causal networks. Basically, we want to see how the effects of gene expression propagate across different cell types to ultimately cause disease. In this particular example, we have here expression of gene X1 in cell type 1, which causes the expression of the same gene in cell type 3, which causes another gene in cell type 2, which then causes disease. So we're really trying to map across how the molecular mechanisms work across cell types and genes. And the final aspect that I wanted to point out here is the Landmark project, which is going to collect a wide range of data on transcriptomics, proteomics, and metabolomics on Parkinson's disease cases. And the aim is to derive causal inference to move forward in identifying potential targets for Parkinson's. And this is here, the aim of the study is to look at 35 million cells. In contrast to human cell atlas needed 7 years to create 50 million cells. So this is really trying to zoom in and understand how the disease develops using different multi-omics platforms, looking at cell type resolution. So this by Professor Mike Johnson. Most of the computational work has been done by Alex Haglund. He has been driving this project. And also I want to thank all of the patients and families who so generously donated the brain samples. And yeah, the preprint is on bioRxiv. I'm happy to take any questions.
Statistical power, how many samples is needed for such an analysis? You know, how many samples at least could it be done? And does power like lead to any false positive or false negative results?
You mean power with respect to the single cell data or to the outcome data?
Well, so how many single cell data should you have to be able to apply this method? If you have 2 small samples, would that lead to?
You know, it's a very good question, and it really depends on what is the type of heritability of the single cell data you're looking at. And what we find is by the decomposition into different cell types, we have a much the heritability estimates get sharper and better. So we have higher F statistics and stronger instruments. So in general, yes, we are really able to get a better hold on the gene expression by doing the single cell experiments. So yes, thinking of other big phenotype, big consortia like EQTL Gen consortium, which has like 10,000 individuals, there's this trade-off. So yeah, I think for many traits, it makes sense to look at this particular single cell data with the cell type resolution. But yet again, power is an issue, and you need to address this by power calculations to see if you're really able to deal with this. Yes.
Thanks.
Yes.
So all the different cell types were really interesting.
Yeah.
The big multiple testing thing.
Yes.
Yes, you deal with that.
So yes, we use an FDR correction for the EQTL calling. That's of course adjusted for.
Thank you.
George?
Here it's one of the largest, and also I think it's kind of reflected here in the number of EQTLs that we get. Actually, astrocytes is not the largest as excitatory neurons. This is the largest number of hits. While microglia, which is like very relevant for Alzheimer's disease, is quite actually a small fraction, but still one of our key findings was actually in microglia. So just the number here says how many we would have available as instruments, but it doesn't say anything about the later findings.
Hi, Verena, I had a question. So we know in GTEx, for many cis-EQTLs, there's not specificity to specific tissues, meaning cis-EQTL can be an EQTL for the same gene across many tissues. To what extent do you see something similar with single cells within the brain?
That's extremely
sorry,
good question. So this is EQTL discovery, so unique versus total. So this is already showing, is it unique to one single cell type or to more than, and you see that there is quite a big number of things overlapping. That's why I think methodological development, as I've shown here, would be super relevant, and not just for the single cell work, also for GTEC, as you mentioned, just to see, is there a crosstalk, and how does expression in one tissue affect another, and how does it propagate? So that's like the thing that
Compare the GTEx.
Alex.
Yeah, so thanks. So I think we did an initial comparison to GTEx, but our comparison in the preprint has been done to the MetaBrain, which is much more relevant and much more better powered for this comparison.
Thank you. I mean, it's fantastic work, really exciting. I was wondering how so there's was published last year a set of ex vivo EQTLs. So these are from sort of fresh tissue, and I imagine you've been working with post-mortem samples, and I was just wondering how your ex vivo EQTLs compare. You won't have the disease status, so it's not the ideal set that you'd like, but I was just wondering how the EQTLs you're discovering as kind of as causally related to the disease, how they map onto the ex vivo EQTLs that were published in the PFC. I think it was from Phil Yeager's lab. Have you looked at that? I don't know if it's called the I didn't hear it was the Chan Zuckerberg thing. I didn't hear it was being called the MetaBrain, but it was a set of ex vivo EQTLs that were not disease associated, and they were published, I think, last year. But it might be good to compare
it
it
would
to.
be
Yeah.
very interesting to look at this exactly because we talk about post-mortem, so we don't know what we lose in the process of freezing the brains. And I mean, that's one of the limitations of the study, but that's currently how we can generate this sample size. That's a very good point. So we did control for age as one of the covariates in the EQTL calling, but that, I guess,
when we run the EQTL discovery, we do control for all the available clinical covariates, such as age, sex, postmortem interval, but we also do PCA modeling and include the PCs as potential additional technical covariates. So we're hoping that in doing so, we do capture age, but obviously that's assuming that age has a linear effect on the association, and there's probably some underlying complexities. We haven't performed the modeling specifically on age, so that's something we'd like to.
Yeah, exactly. It's a very good aspect to see how much you changes, and I suspect it will not be linear.